Thank you for the exclusive talk.
All right.
Yeah, okay, yeah. I can start.
And I will share my screen. Can you approve? Cool. And can everyone see? Okay, cool. And thank you all for inviting me to talk. Um, so I'm not a traditional motor control researcher. I'm a psychologist, and what interests me about motor control isn't just the process by which we control movement, but the mismatch between the complexity of that process and the simplicity with which we experience our own movements. So, based on our best theories of how biological motor control works, you minimally need some control policy, or we'll call that an inverse dynamics model, uh, that allows us to select muscle movements that will achieve particular outcomes. But we don't know the current state of our body, so we also, because sensory transduction delays mean we only receive information about our current state at a delay. So we also need a forward dynamics model. We need to be able to predict future states from actions we take now. We need to represent our motor goals, which may be abstract or they may be concrete sensory targets. We have to represent sensory feedback. We compare our sensory targets to sensory feedback, which might result in a prediction error. And because the body has so many degrees of freedom, all of these representations are very high dimensional. And each time I perform even a relatively simple movement, my brain effectively has to solve a very complex biomechanical control problem. And yet, uh, when I reach out to take a sip of my water, uh, my experience of doing so is that I just do it. It's very simple. And that leads to this notion that of this vast array of representations that we have internally in our nervous system to support motor control, only a small subset of them actually impinge upon our conscious awareness of movement and result in these things like the feeling when I reach out and grab something that I did that. That was me. Uh, and people have discussed topics like do we have free will? How does that work for thousands of years? And a lot of our folk intuitions about that type of belief comes from our just day-to-day experience of I did that when I move my hand or something else. Uh, this is sort of a basic facet of human self-awareness, and so if we want a scientific understanding of conscious awareness, we need to grapple with this relationship between motor control and motor awareness. Uh, but it's also a very practical question because when, say, the sense of agency becomes misaligned from more objective notions of control, this can be very distressing. A number of motor and psychiatric disorders suffer from this sort of problem. You can think of exotic disorders like alien hand syndrome, where people actually believe that their hand is being controlled by an external agent. But also somewhat more prevalent psychiatric disorders. You can think of pathological voice hearing and schizophrenia. Your brain is generating this experience of hearing a voice, but you don't recognize it as something you're causing. You recognize it as something that's coming from outside of you. Uh, Similarly, in any sort of embodied interface design, whether that be like an avatar in virtual reality or, say, a prosthetic, uh, it's worth considering that even if the user can technically control something uh, in the control theoretic sense, that doesn't necessarily mean that they will have the subjective experience that they can control it. And the flip side is perhaps an even bigger issue as we approach a future in which someone like a paraplegic patient might interface with the world primarily through something like a brain-computer interface, that people might think they're causing things that aren't actually caused, as regulators begin to take interest in observing or in enforcing that these types of interfaces preserve user autonomy, users ought to be able to detect when their autonomy are violated. Uh, but we are, in fact, even people with healthy nervous systems are, in fact, quite imperfect judges of what we did and we did not cause, or what we did or did not intend. And there's really a whole literature that I could draw on to pick out findings to try to convince you that this is the case, that we will sometimes take credit for things we did not cause. But this is myotalks, so let's talk about muscle movement. So sort of near the beginning of my PhD, I saw the senior author on this computer-human interaction paper, Pedro Lopez, talk about a user study he did in which he was trying to sort of speed up people's reaction times by using electrical muscle stimulation uh, to move a user's muscle before they would have naturally moved their muscle on their own. And he got this sort of surprising finding that people would claim that they experienced a high sense of agency for these movements that they didn't actually cause. And I was sort of skeptical of this finding, and Pedro was very open to my skepticism. And so we sort of put our heads together and figured out how we could get to the bottom of what's really going on here, and maybe convince me that this is actually what's happening. So we we developed this forced choice paradigm in which participants completed a cue response reaction time task. We would say go, and they would have to press a button as quickly as possible. But the twist is that we said if you didn't press this fast enough, or you don't press this fast enough, we are going to use electrical stimulation to move your muscle for you and press the button before you do. And so after each trial, we would ask them, to the best of your knowledge, did you cause the muscle movement that resulted in the button press, or did we cause the movement that resulted in the button press through electrical stimulation? And so we used a Bayesian optimization procedure to sort of learn this psychometric function of, at each latency or stimulation latency, the probability that they would respond that they caused the movement versus that we caused the movement. And so after each trial, we update this model and we try to apply stimulation right at that 50-50 threshold so we can get these distributions of stimulation latencies where that are highly overlapping, but subjects are differing trial to trial and whether they feel that they caused the movement or not. And so the question is, when is this threshold at which they think that they caused half of movements? And the answer is actually like way, way before they would actually be able to move. So we measure response times when the button is actually pressed in the stimulation block that are inhumanly fast. Human reaction times kind of cap out at around 190 milliseconds. And when we plot the stimulation latency against the reaction time, we get essentially a line indicating that, yes, our electrical stimulation is causing all of these movements, except kind of at the beginning of the experiment before the optimization procedure has done its job. But people do report pretty confidently that they were the ones causing the movements half of the time. And you might be concerned still, like I was, that okay, maybe what's happening is the subject has started to move their arm, but what the electrical stimulation did was speed it up. And we can sort of address that issue if we swap out direct stimulation of the muscle with direct stimulation of motor cortex using transcranial magnetic stimulation and eliciting muscle movement that way. Then we can measure when the earliest EMG response occurs. And this data was collected not by me, but by Yudai Tanaka. Actually, I think this particular subject is me, but this works. When we line up the EMG recordings to the time of the TMS pulse, we can see that on almost every trial, the onset of EMG activity is reliably about 20 milliseconds, the downward latency of the corticospinal tract from the time that we stimulate motor cortex. And you can kind of pick out the trials in which that's not the case by eye. And nonetheless, our subjects, this time me, uh, it works on me even though I know what's happening, are reporting that they have caused 50% of the trials. So now you might be concerned, well, it's 50-50. That's essentially a coin toss. Maybe they're just guessing, or maybe this is a response bias. But when we measure the brain response using scalp electrodes or EEG to the electrical stimulation, within 100 milliseconds of the electrical stimulation, we're already able to decode above chance whether they're going to tell them or whether they're going to tell us that the movement was caused by them or the movement was caused by us. And again, entering into this analysis is only movements that we're confident were caused by the electrical stimulation, not by the subject.
John,
And in fact, yeah,
can I ask one question like on the experimental paradigm before you move into analysis to
yeah,
just
absolutely.
I understand the previous slide like
Hmm.
yes.
Yeah,
Um so what's happening is that you are going to uh go and then you are going to ask the participant to press the button, and then sometimes like you do chance like TMS earlier and sometimes you did late and then you ask if like that's caused by them. Is that what you did?
yes, yeah. So at the beginning of the experiment, we sort of know what their reaction time is on average, and so we'll apply stimulation just kind of a little before that. And then if they're responding that they caused it more than 50% of the time, we kind of shift the timing of the stimulation until they're only responding that they caused it 50% of the time. As it happens, within once the adaptive procedure kicks in, uh, they're really only for like the first 10 trials beating the stimulator like at all. So when we tally up the how many of the trials follow this linear relationship, where the button press was clearly caused by the muscle stimulation, it's like less than 5% of the trials uh, that the subject actually caused themselves. They beat the muscle stimulator very rarely. Uh, and we can just throw out those trials because they're easy to detect.
I see. I like I'm trying to at least it's a very complex design. I'm trying to understand it. So basically, you like so they have a reaction time, and then you always try to stimulate before the reaction time.
Yes.
Before their linear reaction time, 50 milliseconds.
Yes. Yes. It ends up being that we can uh yeah, move up their reaction times by or rather get their arm to move about 40 milliseconds before they normally would move. Uh and still have them think that they caused 50% of these movements.
Oh, I see. Okay, that's very smart. So basically you try have this adaptive function to always try to ask them to press, always try to stimulate because you know the reaction time function of each participant and you always try to stimulate 50 milliseconds before their expected reaction time. So like even though like because of these 50 milliseconds, they will think like they caused it, but it's actually that you stimulate it. And then you are able to filter out which is actually they press it. So all these trials basically is actually you do the simulation, but they always think they cause it. And then you have
Half the
create
time
this.
they think they caused it. But yes, we if we preempted them a little bit less, then that proportion would be higher. So uh we can but we can pretty reliably get these trials that we caused the movement, but they think they caused the movement.
Oh, that's a very smart experimental design. So by doing this, you are able to select a group of brain response which you cause the movement, and but they think they caused the movement by having their reaction times, and then you stimulate 15 milliseconds before that. Yeah, that's
Yeah.
very smart. Yeah, I think I totally understand. Yeah, that's very smart design. Thanks.
Well, thank you. And and whatever process is resulting in their agency judgments, whether or not they think they caused it, is present or begins in the very early sensory response to the muscle stimulation. So this is we know from back in the 80s or something, 70s, the Penfield studies, that people probably aren't actually aware of any sort of like kinesthetic sensation around the median nerve in the forearm, uh, until they're probably not consciously aware of that sensation until almost 200 milliseconds. Well before 100 milliseconds, where we're able to decode whether they're going to respond, that they caused this or not. So uh, the the they they could be guessing, but they're guessing before they're aware of it. Like this seems to be determined by the time the fact that they've moved enters conscious awareness, whether they think that they caused it or not. But that is somewhat an inference. Uh, so back to yeah, Vittorio.
John, just for a quick question. Where was the electrical stimulation placed?
Right on in the EEG experiment where stimulation was applied to the muscle, we put one electrode sort of near the wrist and then one sort of near the elbow. So uh, it's hard to say. You don't get that much specificity of which exact muscles are being stimulated, uh, just because the current path between the two electrodes is unpredictable. But in general, what we were moving was, just because it's a little easier among the fingers to get it to move, the ring finger was pressing the button.
Got it. Thank you.
Yeah, so same muscle groups that would be innervated by the ulnar nerve. All right. You can do the same thing with the index finger. It's just a little trickier to pull off reliably. Or takes longer to place the electrodes. Okay. So we know roughly when this deviation that relates to the sense of agency originates. We know they're not guessing. We know they're actually not causing these muscle movements. It would be desirable to know what sort of representations that support motor control are the ones that are predicting the sense of agency, right? These scalp electrodes weren't actually giving us that much information about the underlying motor processes that are being affected. And so the problem here, of course, is that we're trying to relate two things that we can't directly observe. We do not directly observe the representations that are involved in implementing the control policy in the brain, or the inverse dynamics model. We don't observe the representations of the forward dynamic model that are predicting future states, right? So how a cognitive neuroscientist likes to deal with this problem is to go on this assumption that if two phenomena share a neural correlate, then they likely also share some underlying cognitive process or representation. And what we mean by neural correlate is a area in the brain in which neural activity co-varies with whatever it is that we're interested in the representation of. So if we manipulate sense of agency in the manner we just did, those areas of the brain from which we can predict the sense of agency, we would call a neural correlate. This becomes tricky when we're talking about something really high dimensional, like motor control, where there are so many degrees of freedom and so many different components of the control process. And what I mean by that is that most neuroimaging studies, or traditional cognitive neuroimaging methods, are not really designed to deal with high dimensional phenomena. So a traditional neuroimaging design or functional neuroimaging design is going to be something like I'll have one condition in which I uh, get a subject to use a cognitive process of interest or show them a certain type of stimulus that I'm interested in the representation of, and then one condition where I don't, and then I'll subtract. And we've learned a lot of things from that. Like the image on the left is from a very famous study localizing the fusiform face area, which is a region in the fusiform gyrus that is very highly selective for face processing. It essentially can only be found to be activated on brain scans when the subject is looking at a face. The problem is that it seems like areas that have that kind of functional specification are the exception rather than the rule. They're very rare to find. And when we're doing these, if I have an experimental design where I just have two conditions that I'm comparing, well, two conditions is 1 bit of information. That means that the amount of information per voxel I can get from my experiment is upper bounded at 1 bit. We're not going to use this type of methodology to uncover some process related to motor control unless we artificially reduce the dimensionality of the motor control problem, which would sort of be a traditional approach. But there's another way to do neuroimaging where we can embrace the high dimensionality of the stimulus. And this has become popular really in the last decade. But when I say popular, not that popular. Very few neuroimaging studies take this kind of approach where we relinquish some of our experimental control, sort of do away with these conditioned designs. We use a more naturalistic or ecologically valid stimulus. We quantify the stimulus features in some way, and we ask if these features linearly predict brain activity. And the linearity constraint is important here. So this study played, I guess they're from audiobooks or podcasts. They were audio recordings. And they used the semantic embeddings for the words, like a word-to-vec type thing, to predict brain activity while subjects were listening to these stories. And this was kind of a really surprising result that this ended up predicting so much of the brain, because previously people thought that only this really small patch of the temporal gyrus was involved in semantic processing. But it turned out that when you accounted for the big multidimensional feature space of semantic selectivity, that actually much more of the brain was working together to encode semantic features. The reason the linearity assumption is important is because these semantic embeddings are a really non-linear transformation of the information present in the acoustic signal or the acoustic stimulus actually being played to the subject. By enforcing that your encoding model only use linear relationships, you're saying, is the non-linear way that I have decided to quantify the features of my stimulus, are the features I've chosen the same non-linearities that the brain introduces through neural processing? Have I picked the features that the brain actually cares about? And much more recently, someone did this with a reinforcement learning model and used the activations of a deep Q network as features to predict human brain activity while human subjects are playing Atari games. And that sort of changed the game of this type of design from finding where the brain encodes some information about a stimulus to, oh, now we're actually talking about action selection, how we transform sensory information into action plans. And so when I read this study that I thought, oh, we could actually make a lot of progress in motor control here. If we can take one of these internal models that we think are involved in the implementation of motor control in sort of a computational level description, and we can approximate such a model with a neural network, then we can use the hidden activations of the neural network as features to predict brain activity, to try to identify brain regions that are sort of making those same nonlinear transformations from current state and goal to actual sequences of muscle activity, right? And of course, MyoSuite has really been the enabling thing that has allowed us to do that, because now one can train these models that are actually able to control with some level of biomechanical detail an actual or simulated human limb. And so the the main technical difficulty is in getting humans to do something comparable to the simulation environments in MyoSuite, right? Uh, so we, perused the natural policy gradient baselines in the original MyoSuite release when I came across this, and tried to find something like what's the most interesting thing here that we can get humans to do in the scanner. And I would have loved to do one of the object manipulation tasks, but well, we have all of this wonderful technology for tracking body motion, for tracking objects. Most of it stops working when you put it next to a 3 Tesla magnet. Uh, so we were uh, a little stuck, but we do have a motion tracking glove that can measure joint angles, but it's all optical. And so MRI compatible. And so we had humans do a task that was supposed to mimic sort of the skills learned by a model trained in the Myo hand pose random environment. Uh, so we just showed humans the pictures of hands, said mimic this position, and then they would mimic the position, and they were instructed to hold that until the next target appeared. We presented a new target position every 5 seconds, and they did this for almost 2 hours. So that's what I mean by not a supernaturalistic task, because it's not exactly what I go home and what I'm planning to go home and do tonight is not switch hand poses every 5 seconds for a few hours. But the but because we could measure the joint positions and we could measure the velocities and we knew the targets while people were doing this task, we had all of the same things that are inputs to the natural policy gradient. We just used the pre-trained baseline. And so we could input those features at every moment while subjects are participating in this task. We could extract the activations of the hidden layers, and we could use those features over time as predictors of the bold signal over time. And then we do need to account for the fact that it might just be that it's not the specific nonlinear features learned by the MyoSuite model that are predicting brain activity. It might just be that having any random nonlinear transformation would be helpful. The same sort of notion behind a kernel trick. And so we enforce that prediction of brain activity from the activations of the neural network are linear, but we also have a control model that is just a nonlinear kernel regression. It can learn any sort of relatively simple nonlinear transformation of the input features, and we gave it access to all the same information that we gave the neural network access to. And so our sort of localizer for inverse dynamic representations, and we're just going to call this an inverse dynamics model. Then our localizer is the MyoSuite model, R squared per voxel for predicting each voxel, minus the control model's R squared. Wherever that difference is positive, the MyoSuite model is performing better. we're going to say those are inverse kinematic selective voxels. All right, and just to show you what the fit models look like, any voxel that are and what we've done here is cortex is kind of a folded sheet, and just to help you see the whole brain at once, we're going to unfold and flatten the cortex. And so each of these are models fitted to a single subject. Any voxel that is colored is a voxel that we could predict above chance after correcting for multiple comparisons. And the color is denoting the sensitivity of the predictions of the model to these subsets of the feature space to the neural network. So say a voxel that I've colored in pink is responding primarily to combinations of joint positions and the position error. So it's probably encoding something about the target position. Whereas a voxel that is entirely red is probably only encoding the position of the hand. And just what I want to highlight here, and we'll sort of return to this point later, is that which voxels we're able to predict are pretty similar across subjects, but if you drill down to how I've colored them, what parts of the task space or the feature space are predicting each voxel are really idiosyncratic. It's not the same in every subject, and that ends up being really common in these voxel-wise encoding studies where you're looking at how a high-dimensional feature space is encoded in the brain. We'll sort of get to reasons why, but this is something that I wouldn't be the first person to point out. Now, when we take our MyoSuite models, R squared per voxel, and subtract the control models, the places where, again, after correcting for multiple comparisons, we see that the MyoSuite model most robustly outperforms. That was surprisingly all in visual cortex, or at least that's what our localizer highlighted. When your head is in the MRI coil, you can't actually see your own hand. So that doesn't explain this. Both models had access to, say, like when visual stimuli were presented and what the targets were. So that wasn't necessarily explaining it. It seems like we found evidence that inverse kinematic representations are to some degree represented in visual cortex. And this was a surprise to us. It was not a surprise to everyone I've talked to about it, including our reviewers, because we have known for some time that, say, grasp poses or reach directions can be decoded from early visual cortex in sighted people who have been blindfolded, and in congenitally blind patients. So this doesn't seem to be dependent on visual experience at all. It's just there are representations of the hands in visual cortex, and our results would seem to indicate that they're involved in inverse dynamics or the implementation of a neuromuscular control policy in some way. So now we can go back to our original question, and we had these same participants come back days later, and we had them do that muscle stimulation task I showed you before. We gathered all the trials, almost 1,000 per subject, where the movement was in fact caused by muscle stimulation. Every subject, it ended up that almost exactly 50% of these movements they thought that they caused or reported that they caused, and we decoded these judgments or predicted these judgments from a whole cortex decoder. So what that looks like is we just assign a linear weight to each voxel. and then two mask decoders. So our visuomotor mask is all of the voxels where we were able to predict the activity in that voxel in that session one motor localizer at all. And what a mask decoder means is that they only have access to the voxels inside the mask, so only those get decoding weights. And then our what I'm calling the theory mask is just that small set of voxels in which inverse dynamics, or that we're saying are selective for inverse dynamics, where the MyoSuite model outperformed. And we are able to decode the agency judgments above chance from all of those, indicating or aligning with our hypothesis that neural representations involved in inverse dynamics do impinge upon the sense of agency. Now there's an asterisk here, which is that not all subjects look the same. So instead of doing that analysis at the group level, we can do this localizer just within each subject. And when we do that, I mean, we still only found activity in visual cortex, but where exactly in visual cortex? Here we have ipsilateral and contralateral. Here we just have contralateral to the hand they were using for the task. Here we only have ipsilateral. Where the brain maps look very different. And some of that is a sensitivity issue, I think. So like this patch of cortex between V2 and V3, nominally, the MyoSuite model outperforms in everyone. So probably if we kept collecting more data, we would eventually have the statistical sensitivity to detect that. But I do think these individual differences are meaningful when we use just the voxels where we were able to detect selectivity for the representations of the MyoSuite model in each individual subject, as opposed to in the group average. It really boosts decoding performance for sense of agency. And so much so that the difference between just using the individual theory masks and using the visuomotor mask, so everything that was involved in that motor task before, or even all of cortex, the difference in decoding accuracy between those is no longer statistically significant. So the boost is quite substantial. So why are we getting such sizable individual differences? Why do the selectivity maps of these voxels look so different across subjects, but are so robust within subjects? All of these models are cross-validated. The one answer is that it's just another manifestation of the curse of dimensionality, and that's why you tend to see this type of phenomena in these when you fit these encoding models for high-dimensional stimuli. The more dimensions your, like, say, subjects are all coming from some probability distribution, the more dimensions over which you have to spread probability mass, that's less probability mass that can be concentrated in some volume around the population mean. And so the more dimensions you have, the less any one subject should be expected to look like the population mean. And so plotted here is just the probability mass at the mean of the normal distribution, standard normal distribution, as you add more dimensions. And it's an exponential roll-off. So to some degree, this is expected. But you also have what I think is a more foundational issue with how we do human neuroscience, which is that and I do say human neuroscience as opposed to those using non-human animal models. If I place an electrode in two different brains, I might say, oh, I placed an electrode in motor cortex of this mouse and motor cortex of this mouse. But I would never say that the cell I found in this mouse is the same as the cell I found in this mouse. At that level of granularity, it sounds nonsensical. When we're doing human whole brain neuroimaging, because we can sort of warp brains such that all of the anatomical landmarks align and then average, we're very tempted to do so because we want to generalize from our individual subjects to the population. But if there are subgroups of subjects or subpopulations that, say, in a given voxel show an effect, but also, plus or minus some sampling error, but there are also subjects who don't have an effect in that voxel, plus or minus some sampling error. The true population mean is still going to be a non-zero effect size. And if we collect enough data, our statistical test will be sensitive to that, right? And we'll say that there's an effect in this voxel on average. And that's going to be true even if the proportion of subjects in the population who actually show this effect is very small. And so when we report these group mean maps, we're not, once we have enough data, we're not actually looking at which voxels are most consistent across subjects, or we're actually looking at the brain activity of sort of every type of subject superimposed on top of each other. And I think this is a major challenge in neuroimaging. Like intuitively, the ability to image brain activity non-invasively should have enormous clinical potential, but that clinical potential has not been realized. And I think one of the chief drivers of that is that the results that we report in our studies don't really look like any individual person. And so I'm pleased that it wasn't just the case that the features from MyoSuite models helped us predict brain activity better than just any arbitrary nonlinear feature, but actually at the level of individual subjects, we were able to establish these robust correspondences between the models in the brain, and that these did seem to be sensitive to individual differences. That is going to be a very powerful tool for us going forward, I think. I also think we can make these models a lot better. So we just used the natural policy gradient baseline. And this is the training curve for that. And definitely during training, the model got better at the task, right? It learned something about controlling the hand. But unlike some of the other baselines in that original MyoSuite release, and unlike a lot of models that the MyoSuite community has come up with since, the Myo hand pose random model did not solve the task. Right? It doesn't approach human level performance. So my guess is, or I'd hope, that as we start to use models that approach human level performance, that we're going to be able to predict human brain activity better. But I also don't think, and as we incorporate more, right now we just used a simple controller, right? Goes from state and target to the muscle activity that should generate the desired state. But in biological motor control, we also have, we think we have forward models, we think we have sensory feedback represented, and we weren't really doing that here. So hopefully, as we incorporate more components and we use better models, brain prediction will improve. And so I've been keeping an eye on what the MyoSuite community has been doing, with hopes that we can use it to understand human brain activity better. But I also don't think that even if models and simulation can reach human level performance, that isn't necessarily a guarantee that we're going to be able to predict all human brain activity, or that the representations of these models will resemble brain activity. And we're already seeing hints of that in this first pass. So given that the output of the model we used is muscle activation, you would hope that we're able to predict motor cortex, which is projecting from cortex down the corticospinal tract, more or less directly to the musculature. And our prediction of motor cortex is above chance, but it is also above chance in the control model. And actually, the control model predicts activity in motor cortex better. So while we found evidence of inverse dynamic selectivity in visual cortex, you have to assume that our our approach was not sensitive to all inverse dynamic selective voxels throughout cortex. And there's sort of a predictable reason for that, which is that we know only a small fraction of neurons in motor cortex have activity that resembles EMG, or that resembles the output through the cortical spinal tract. Most motor cortex activity is related more so to the network level dynamics that are necessary for producing that EMG activity, and not the EMG activity itself. So for example, motor cortex needs to be able to engage in motor preparatory activity without actually generating corticospinal output that will result in movement, right? You don't want to move while you're preparing, you want to move while you're moving. And so if we think of motor cortex activity as a population vector, where the components of the vector are the firing rates of all the cells in motor cortex, you'll see during motor preparatory activity, you're moving around in this null space, in which it can sort of vary neural activity without varying corticospinal output. And then when movement starts, you sort of flip up into a different subspace, in which movement does result in corticospinal activity. And the biggest axis of variation of population activity in motor cortex. And remember, when we're doing whole brain imaging, especially non-invasive imaging, all we're able to see is the aggregate of populations of cells. We don't have single cell resolution. The biggest axis of variation is movement onset, which I think our control model learned. That's a simple feature. And so a model that is going to predict brain activity has to capture the network level dynamics, temporal dynamics, that are actually necessary for producing movement. And that's sort of the limits of thinking of the brain as solely a representational system, rather than also a dynamical and a real biological system. And I'd like to move into more naturalistic tasks in the future, or more ecologically valid tasks like object manipulation. Again, there are a lot of problems with doing this stuff in the scanner, but there is such a thing. I don't have this hardware, but I hope to have this hardware soon, as MRI compatible virtual reality. And so I was happy to see at the Myo conference yesterday, people are already doing cool things with MyoSuite and virtual reality. And so my hope is that that's going to help us clear that hurdle, and we can start studying interesting, ecologically valid, and relevant tasks. And I think if we keep going with this approach, we're going to be able to answer a lot of fundamental questions about how the brain represents the body. Like, do all these individual differences we see in the brain correspond to, or how do they relate to individual differences in anatomy? If we use anatomically specific models rather than just use the one size fits all model to predict brain activity, is that going to make the individual differences we see in the brain disappear? Or are we going to see more individual differences? And I really don't know. That's kind of a foundational question about the organization of the sensory motor system in the brain. And then once you start talking about perturbations, whether that be like an experimental manipulation of sensory feedback, introducing a delay, or a particular type of perturbation that's meant to be assistive, like an exoskeleton, how does that change the encoding of movement in the brain? And of course, people in the MyoSuite community are already doing fabulous work on this type of thing. And I would love to collaborate with anyone who's interested in using neural measurements to inform that kind of work, or having that kind of work inform neural measurements. And I'm very open to collaborations. This work was really meant to be a proof of concept that we could establish these robust correlations between models trained in simulation with no human data at all and actual human data observed in the scanner. We were sort of approaching the end of a grant, and I was like, wouldn't this be cool to have as pilot data for future grants? And it sort of ended up being so interesting that I thought it would work as a standalone publication. And luckily, some editors and some some reviewers agreed with me, and this work was just accepted to the Journal of Neuroscience. But it was really a pilot study. This is the beginning, not the end product. I'm excited to see where this goes. Speaking of collaborations, I would like to thank my collaborators as well as my funders, without whom this work wouldn't have been possible. Hmm.
Fantastic. Thanks, John. Like I think it's the best work I have seen in in the whole year in neuroscience. It's yeah, it's really you use MyoSuite to unlock the high dimensionality of the human behaviors, and you're able to show the person like the individual difference between the brain. So you're able to build personalized brain models, like because when the dimensionality of behavior become unlocked by MyoSuite, the brain difference, the individual brain difference is also high dimensional, also get unlocked by this approach. And then now you are actually going to have clinical brain, individual brain model that's possible. I think that's extremely exciting. I believe Vittorio and Vikash have some question to ask.
Yeah, thanks. Go ahead.
Yes, John, very interesting work. I have a few questions. Maybe I just start with a couple, then I go on after others. So you use the neural networks to predict the brain activities. Which layers of the neural network did you use, and did you observe any difference using different layers of the network?
Well, the natural policy gradient baseline, the model just had two hidden layers in the output. So we used two hidden layers in the output, which seemed we didn't really look at differences in layer by layer, uh, just because the network itself is so shallow, which differs from other work using neural networks as an encoding model, where they tend to use like uh, ResNet or AlexNet or something for vision applications, right? The transformation layer by layer seems to correspond pretty nicely to the transfer to like the first layer resembles early visual cortex more, and later layers will, uh, will resemble other areas of the ventral visual stream. Like that hierarchy seems to be consistent across the neural network models and across the like known organization of the human visual system. That's because these types of networks are more shallow. That was sort of less interest here, but I think in the long term is what makes using reinforcement learning models more interesting to me. Because the difference between, say, an RL model trained with uh, direct policy optimization versus like model-based reinforcement learning, those correspond to meaningful like descriptions about the organization of sensory motor systems that are actually the same types of descriptions that we use when we're talking about like biological motor control theories. And so by switching out, now I'm using a model-based version versus now I'm using direct policy optimization versus now I'm using actor critic. We can think of these as testing different theories of motor control on the basis of how well they predict brain activity. So I think I'm less interested in sort of going layer by layer and trying to interpret what the neural network's doing, which is kind of explaining a black box with a black box, as there's this super elegant thing about deep reinforcement learning where it's not all a black box. You did specify how all the components click together. in a way that when you see similar work in computer vision, they don't have that going. Yeah. Yeah.
Okay, great talk, John. I think I have more questions than answers at this
So do
as well.
I.
I'm curious about a couple of like maybe more fundamental questions. So let me start by a few. Is you know clouds, like people see even like patterns in clouds, right?
Mhm.
So if you know what you're looking for, you'll find it in clouds as well. Right. My question is, did you think of like somehow showing a negative result that, okay, this doesn't happen with all networks? It happens particularly with like, okay, the networks that were trained for reaching. Like, for example, like I would envision things like taking a network that was doing some hand manipulation one and then do reaching with that and show that it's worse off than the reaching one at least. Or take a random network and say that it has zero correlation at all. So I'm curious if you attempted any of those things.
Yeah. I mean, we've tried a random network, and I think the best way to generate a null distribution in the future to compare like a trained model against is probably just, comparing it to like 1,000 or so uh, randomly initialized networks. Here, it was easier. We just used a kernel regression using the same input features. So we're saying like, oh, the nonlinearities of that the neural network learned in the MyoSuite simulation uh, are better than the nonlinearities that like could have just been learned from brute force this human data set. Uh, I think in the in the future, yeah, I'd like to explore more uh like uh like after some sensory motor manipulation, the model that you've trained, say, pre-surgery in the simulated tendon transfer surgery, uh is going to do very poorly if you uh try to have it generalize to post tendon transfer without retraining, right? So one question is, do people who have had some sort of surgery or use some sort of assistive device, do they look more like a MyoSuite model that is trained on something similar to their current sensory motor context than the model that's trained on something that is uh, more like a baseline like anatomy of the hand,
Mhm.
right? That's an interesting area for future research and a direction I definitely want to go. Uh, but yes, yeah, I think what you highlighted is definitely the reason we incorporated a control model rather than just saying like, oh, we were able to predict all of these voxels. We were able to predict all of these voxels, but the control model could too. Uh, the only place where the MyoSuite model seemed to really specifically improve prediction was in visual cortex. Uh, but we also know that's not capturing all inverse dynamics in the brain, right? So we want to improve that.
Yeah. So some of like these ideas, I think I like quite a bit. Expanding a bit on that, let's just say I would also be interested, and I think it'll be very interesting to run a few ablation studies here, where we can try to correlate activities with a few different kind of representations. And those representations, for example, can be that the representations learned by the network that was indeed doing the reaching tasks. So it's strongly correlated with what you asked the users to do, number one. Number two, maybe instead of pose reaching, we can do velocity tracking tasks. We can do, I don't know, like some manipulation tasks, or like maybe just a single trajectory tracking rather than a random trajectory track. So you see the direction or the dimension I'm trying to probe, right? We try to stay closer to the task that you ask the human to do, and then slowly create other networks that are farther and farther away from the tasks that you ask them to do, and also the dimension of generality, that instead of like asking to do a lot of those reaching movements, I always ask them to like reach for like 90-degree bent fingers, right? And see if the relative correlation decays or remains the same with it. That could be very interesting thing to know. The second interesting thing to know is what we have learned over different experiments, like maybe in the machine learning sense, less validated right now, in the machine learning sense, is that generalization emerges if you expose the agent to a wide variety of environmental circumstances, that you cannot find local minimas. You really have to find solutions that generalize. And I think human learning, my intuition at least about human learning is somewhat similar. That as a baby, you find esoteric way of walking, but then as you try to do different things and then like, play soccer and stuff, you start harmonizing that, or all those esoteric way of like walking sort of dies down into like something much more generalizable, right? So I think I'm also kind of curious that if we take some of our multitask policy networks, for example, the MyoDex networks,
Mhm.
see better correlations with respect to those networks, which has been exposed to much richer variety of tasks than these networks, which were just like trained for posing, right? I'm asking maybe a lot of open questions, but that's also communicating my excitement of like which other directions are worthwhile looking into and could be super interesting to know more about them. Yeah.
Yeah, and definitely I definitely had plans to use MyoDex or take a model like MyoDex that's able to manipulate a lot of different object types and compare it to say models that are trained for each individual object that the subject is manipulating. Uh, and I expect that uh, that sort of as you suggested, there will be representations that are task specific, right? If I'm velocity tracking, then I'll be better predicted by a velocity tracking model. But there will also be representations that are just generally useful for motor control, right? Uh, and using generalist models compared to these task specific models could allow us to dissociate between the types of representations that are present in the brain or the types of sensory motor systems that come online for both these sort of generalist skills and these really task specific skills. Um, so yeah, I think there's a lot of potential here to answer uh,
Yeah.
pretty foundational questions. Of course,
Absolutely. Absolutely. As you probe more, if you need some help from our side on these things, keep us in the loop. I think this is very exciting. I will have one more follow-up maybe question, and then I'll pass it on to Vittorio. He has more questions. So the same way I think we talked about, one axis of probing is the task axis, right? Like we can do multitask, single task, like much correlated task to what you ask the user. Or you can flip the user saying that like, okay, it was trained for this task, but you do different tasks and see, let me see if the same network representations correlated with all kind of tasks that you are trying to do, right? So that's one dimension. I think we had a chat about that. The other dimension is now let's go towards much more machine learning models. Let's just say you already had your recordings from the human that was doing like all of these reaching tasks. And now I want to train different networks that were doing the same, random reaching task. And each network is trained in a much very different way. Some are trained using model-based reinforcement learning. Some are trained with actor critic. Some are trained with sparsification objectives. Some are trained with maybe let's just say, for a lack of a better word, like energy minimization terms in there. So there is a lot of different ways that we can train the same neural network to do the same task, right? And that axis is also like worth exploring to see which objective correlates or improves the correlation. And if we can find something interesting over there, then there is a hypothesis that builds up is possibly we are trying to find or the objectives the brain is using to like do the same thing. Obviously, like we call like there's optimal control theory of brain. There is like always like jerk minimization theory. There is energy preservation theories, and all of these theories can be embedded in the network itself during the training process. And if the correlation improves, then I think that's also a really good validation that comes up, right? Yeah.
Yeah, so I'm definitely thinking about collecting these sort of large human neuroimaging data sets that we can then use as sort of a benchmark. Like imagine we had neuroimaging data from humans doing the same task from one of the Mayo challenges, and instead of just benchmarking all of the submissions to Mayo challenge on how well they do at the task, well, we could also benchmark them on how well their representations predict brain activity. And I that yeah, the being able to both rank models or look at what types of training features predict brain activity or improve prediction of brain activity, I think should definitely tell us something about the brain, what the brain is doing. But you could also find like, oh, these two types of training like predict this or improve prediction for the same set of voxels, right? But
Mhm.
not one, but it's either or, like not one predicts it more than the other. You could discover something where the brain sees two components of a task as the same thing that we wouldn't necessarily have expected. Uh, the same way that like the hippocampus is used for long-term memory, uh, but is also used for spatial navigation,
Right.
and that like doesn't intuitively make sense to us, but when you dig into it, in fact, it seems to be using the same sort of neural mechanisms for both types of tasks. It found that like the brain capitalizes on this computational similarity that we wouldn't have designed from first principles. So I think there's that opportunity for exchange where we can use all of these models that the MyoSuite community is building to learn about what the brain is doing. But it also might suggest these unexpected avenues for or connections between different models.
Absolutely. An opportunity I see in front of us over here is if the data that you're collecting is shareable, then we can potentially try to host it right next to all of these environments and then, pose it as a challenge or an open questions in our GitHub forums or like on our web pages that people should look into. But I don't know if the data that you have is shareable or not, so I'll let you be the judge of that. But
Ready share. It's on Open Neuro. There's a link in our paper.
excellent.
But I can also share it on whatever or a link to it on whatever other platform. Yeah.
Excellent. All right, Vittoria, go for it.
Awesome, John. I think that your question to start with was really inspiring for me to put even on larger picture. So any time that we talk about sensory motor control, is really the sensory part is one of the most complex feature. And one thing that we have been trying for long time now to disentangle is the senses, how they interact in terms of proprioception, visual. Now, probably you're familiar with the literature trying to disentangle execution from observation and how those things come together in the imagining moment. Because the imagination now decouples the execution from the observation in a layer that is more abstract. Your layer now has a fourth dimension to this picture, which makes everything even more interesting. And what we have learned a lot on the interaction between the motor imagination is about also motor suppression. How you try to suppress the fact that you're observing something is not triggering, since we have some evidence in physiology, actually in experimental neuroscience, with single neuron recording mirror neurons and neurons that are related to the execution and observation of action, that you have the same neural substrate that will be respective to both. And when you come to the sense of agents, this actually becomes the most important part. Those are really exactly the ones that are responsible for that sense of movement, sense of direction. So this is, for me, is now the most interesting part. So we have all these dimensions that we can decouple and we can simulate. And that's the part where I think that many of the things that you have been observing might get an explanation, because you do not just execute, you don't just observe, you execute and sense, you can execute without sensing. So I was wondering if you start thinking about this direction of how those different components come together. And also, I mean, your experiment basically is a way to disentangle those, because now you can just cut and slice with the current paradigm across all these size and dimensions.
Yeah. Yeah. I mean, so that is the exciting thing, especially as we start I just the model we used, I just fed joint angles and stuff as given, right? But we don't get proprioceptive like direct proprioceptive input of joint angles, right? In the brain, we get muscle tendon lengths, right? Uh, and we don't, and that's not the only way we sense our hand. We also get the visual feedback, and uh, so one of the things I'm excited about in the future, like incorporating virtual reality, is incorporating the visual sense and better, doing the inverse kinematics to get the joint angles and using more realistic models in terms of the inputs, is we can really capture this full sensory motor loop in the way that's never been done in human neuroimaging before. Uh, and the conscious awareness component is is as a long-term goal, right? We talk about reinforcement learning agents and training agents, but the notion of agency is, or what makes something really an agent as opposed to a program, or whether a program could be an agent, right? Those are all really wide open questions that no one really has an answer to. Uh, Measuring the sense of agency and what types of representations at what point in the sensory motor loop are predictive of the sense of agency will at least be able to tell us that, well, hey, uh, which of these representations are related to our experience of being an agent? Now, that's a very different question than like a formal definition of an agent, right? Uh, it might be that our experience of being an agent is all epiphenomenal, uh, or it might be that will point us to this is what distinguishes something that's autonomous from something that's just a program, right? Uh,
If
so
I
there,
might interject on this, I think that this is exactly one of the key aspects of what you have been showing, that there is a difference between what you sense as your own activity. We have done these experiments in the past, like reproducing movements that you have done and what movements are done by others. Now, the movements that you have done, for the peculiarity of the movement itself, you can recognize that is yours. Point of view can be kinematics or dynamics. Same movement of others, they have some signature differences, and those are correlates of the neural activity that you observe. Now, excel the sense of agency is the key point there. And that's why I feel like is exactly the tip of the iceberg to close the loop of all this understanding.
yes, absolutely. Uh, this is it's a very exciting place to be right now, just from there's a fundamental philosophical, like, we can answer or get at questions that people have been asking for thousands of years from a new angle, right? But then also, I do think there are very real uh, applications to this type of work, uh, and so this is, I think, a really productive avenue for neuroscience, and MyoSuite has really enabled this to uh, or enabled me to, I think, take this space in a direction that people at this intersection between consciousness and motor control have really not been able to get into yet.
Yes, fantastic to get this started. I feel super excited about it.
Yeah, and thank you all for all and the rest of the MyoSuite community and all the work you've done on this tool.
Can I ask one question, like on the application side and the consciousness side? So
Yeah.
one application is neurorehabilitation, like stroke rehabilitations, like where neuroimaging or personalized neuroimaging really is critical because like when for every stroke patient, they have a different brain, right? For every stroke patient, like their brain is different from each other. Like none of the stroke patients share the same damage. So when you do fMRI, like the brain literally look different from each other. It's personalized and it's individualized. And one problem in the past is that this kind of individual difference between stroke patients was never treated in a sense because there's not enough data, and then like there's standard stroke task like behavioral tasks, and then they do and then they put the stroke patient into scanner, and then they try to overlap the damage area, which like and then like they have like 10 patients, they all have different damage, and then they have a shared damage area, and then the stroke rehabilitation or neuro stroke rehabilitation research trying to make some claims like, oh, they all lost like this area and then it's related to this motor function.
Mhm.
So I'm trying to ask, now given the high dimensionality of the all the control policy, all the reinforcement learning, and like all these different kind of neural networks you are enabled, and you already observe the individual difference between the healthy patients. So I imagine if you put like 20 stroke patients like into the scanner, and their motor dysfunction are all individualized, and that have never been accounted in the past, like with the like neural rehabilitation that's being practiced in the like in like nowadays. Would you imagine that I put this stroke patient in the scanner, and then I'm able to do some clinical applications, and then I'm able to have really good neural networks that's going to model particularly these stroke patients and his motor dysfunction. Because like you see, like people, some patients, like their index finger, this dimensionality just gone away. But some people like this dimensionality, they go away. Like so, like there's a lot of like even in the manual like hand dexterity task, like stroke patient differentiate. Would you imagine that like now we are finally allowed, given the availability of high dimensionality of the MyoSuite, individual difference with a personalized neuroimaging, and then like every now stroke patient with different brain damage, and then now we can actually have a personalized model on the brain level and the control level, and also on the behavioral level, which that this can be unlocked.
Yeah. So like you can imagine if we have this type of mapping between a MyoSuite model and the brain, that if we took one of these healthy brains, we said, look what's encoded here. If I were to lesion here, and we don't have to lesion here, we can use like magnetic stimulation to selectively disrupt that part of cortex. Uh, would we observe the something as if the or would we observe a change to behavior as if we had lesioned uh, the corresponding part of the MyoSuite model, right? Uh, and so you can imagine that when you're working with a stroke patient, you could fit this sort of model and say, okay, which parts, instead of using the model to predict the brain, what if we use the brain to predict the MyoSuite model? Uh, and say there are some activations in the hidden layers that we're very bad at predicting. So we remove those activations or like knockout from the model. Does the behavior of the model then become like the stroke patient? And if so, then what manipulations can we do in simulation that restore behavior of the model to something that looks more healthy or normative? And uh, I think there's a lot of potential with that sort of approach, in which we can maybe use neural data to create more tailored strategies for rehabilitation. Or say you're using an assistive device that has its own controller, right? So like an active exoskeleton. Uh, then then well, the active exoskeleton you would like to adapt to the user, but you also want the user to be able to adapt to the adaptive skeleton or to the adapt or to the exoskeleton. Uh, can we use say something like a brain-computer interface to let the exoskeleton know this is the current state of the user's control policy, uh, and guide them in sort of a game theoretic way to a better ultimate outcome, right? Uh, so these are sort of some of them are far-fetched now, but also not ridiculously far-fetched
Yeah.
that we could use neuroimaging uh, to create these better outcomes in the long run.
And also another mystery in neurorehabilitation is like in stroke is that when you get the brain damage, what happened in the 3 months, which they call the acute stroke like period. Like the first 3 months when you lost all the when you like when you just get stroke, like you still have some kind of policy residual, and after 3 months you lost all those dimensionality. And it's not like I believe it's until today it's not like the neuroscientists don't know what happened like during that 3 months. Like some kind of rewiring or whatever is going on. But like this, like and then like essentially, I don't think we don't know what happened. But behaviorally, like we see loss of function, and then like brain imaging, we see loss of lesions, right? And then something happened in the brain, like during that 3 months, and then stabilize, and then you become like chronic patients. So I would ask, like, do you think that's something that's really you can allow to explore?
Yeah, absolutely. There's a lot of potential for doing this longitudinally. Uh, so both both say in someone who's experienced a brain injury, you should expect a good amount of neuroplasticity that I think we now have the means to track if we're using this kind of high dimensional model. But even in a healthy person, I mean, we have some expectation that that the that like I did 2 sessions with these subjects, right? And where we localized the inverse dynamic representations were seemed to be pretty good at telling us where we'd be able to decode sense of agency in a subject specific way, like days later. So it seems like individual differences are preserved across days, which isn't always true with neuroimaging findings. Decoding across sessions in the same subject can be quite poor. Uh, but we have no expectations about what would happen if we brought in the same subject for a year. I did bring in one of these subjects. It's not in the paper. I haven't analyzed the data yet. Uh, but we did collect data from one of these subjects, the one who was kind of most of an outlier, uh, like 3 months after his initial session. Uh, and so the uh, I'm interested to see if the encoding model looks the same on, that session 3 months later as it did 3 months earlier. But by and large, uh, we don't really know, even though we know on a relatively short time span or within a week that these individual specific encodings seem consistent or robust. On a longer time horizon, we have no idea. Someone's map could look totally different if we collected uh, data from them again a year from now.
Yeah, that longitudinal study of the brain like plastic is extremely exciting. And then now you are finally able to catch that high dimensional space. Yeah. Great talk. I think we actually take 1.5 hours. I'm going to stop recording now and then put it. Yeah.